Okay, right. So, hello. Welcome to my video about glycolysis. I am planning to simply cover some basics that you might be, asked to learn during your university course or are just curious about what it means. Obviously, there's so much more to research online about the topic, but I'm here to cover just some, foundations of glycolysis. But obviously, please do some further reading onto the actual topic to reinforce your understanding. So basically, this is how you spell glycolysis. But before moving on to the actual, content of it, I want to firstly analyze the words glycolysis. Now, we can split glycolysis into two words, which are glyco and lysis. Glyco comes from the Greek word glykis, and so forgive me if I don't pronounce that correctly, but it's so this is how you spell glykis. And we have a lysis, which means split. Now, glycus means sweet, and therefore, as basically this is kind of like describing your carbohydrates, your sugars that you have in your bodies, etc. And therefore, combining both the word sweet and split, we're talking about splitting your carbohydrates. And as you might remember from some basics of biology, is basically like polysaccharides basically being split into two kind of like components. So your smaller polysaccharides or disaccharides, or for example monosaccharides, for example. And it's basically involves some of that, but it's a bit more complex than that. But before going into delving into the main content of this video, I want to first talk about the importance of glycolysis. So what's the importance of glycolysis? Now, why do we need glycolysis? Well, both prokaryotes and eukaryotes use this process of glycolysis, and it's essentially a fundamental metabolic pathway to produce energy, which is in the form of ATP over here, and also produces other molecules which are called intermediates, which are needed in other processes. Now, furthermore, some cells in our bodies don't have mitochondria, or they might be under anaerobic conditions. An example of this would be, for example, your red blood cells. So you have got the diagram here of a red blood cell, and essentially this doesn't have a mitochondria. And its function, as you might know, is to transport oxygen around into the designated places, into the target cells. Now, essentially, you don't want this red blood cell to use the oxygen that it carries. It's a transporter, and preferably we don't want it to use the oxygen that it's carrying, and it's meant to be designated to other cells to use it, not itself. So that's why we have glycolysis, which essentially will allow it to produce some ATP, some energy, without it using the oxygen. And also it's very useful actually during conditions of anaerobic conditions where there's not much oxygen available in the surroundings. So it basically means that they can still produce, so through the process of glycolysis, they can still produce energy in the absence of oxygen and in these conditions. Now here is a very simplified diagram of glycolysis. So where we basically start with a 6-carbon molecule, which basically there's an ATP being converted to ATP after we have taken taken a phosphate from each ATP molecule. And we use this to phosphorylate this 6-carbon molecule to produce this. Okay. After that, we will split this molecule in half, producing these two 3-carbon molecules phosphorylated, and then we will take another phosphate, not from the ATP as you've seen before, but actually from the cell solution. So this comes in, phosphorylates this molecule again on both molecules, and then we will have an NAD plus coming in, which would then be reduced into NADH and H plus, which is your proton, and also you will get again an ADP in being converted to ATP. And because this happens twice, because we got 2 3-carbon molecules, it will get so we will produce 4 ATPs. So recapping this so far, we have added 2 ATPs going in, and we have 4 ATPs going out. So therefore, overall, it's a net production of 2 ATP molecules. At the end, we will produce pyruvate, which are very important molecules, and it's linked to other metabolic pathways, which are, Krebs cycle, which we'll cover in later videos. So now we got to the actual, actual main part of the glycolysis, which is more kind of like in depth. It's not talking about simplified version of it, but more molecular, in the molecular level. Now, so step one. So we have basically your glucose, and this is the molecular structure of glucose. Now, this basically produces your glucose 6-phosphate, which essentially I've also highlighted some the actual difference between these two molecules. So what I actually like to do, which helps me to remember these steps, is to basically have it note the difference between the initial molecule and the final molecule. As you can see, the initial molecule here in carbon 6, so 1, 2, 3, 4, 5, 6, essentially has an OH at the end, and here is an OP. So we have phosphorylated this part, this carbon 6, using the enzyme hexokinase. Now, as you can see from the arrow here, it basically we have introduced an ATP going inside into the process and then leaving as ATP. In other words, we have taken a phosphate or dephosphorylated the ATP to make ATP, which has essentially, which the hexokinase has phosphorylated this glucose molecule, specifically in carbon 6. Now, just to go back a couple of steps back, I want to highlight two forms of enzymes, two types of enzymes that we will be using throughout this entire explanation. Now we have kinase enzymes, and phosphatase enzymes. Now, kinase enzymes, what they do is that they're in charge of phosphorylating molecules, whereas your phosphatase enzymes, they're basically in charge of dephosphorylating these molecules. So in this case, actually, this is actually written in here. This is a kinase enzyme, hexokinase. Essentially, this hexokinase will phosphorylate this carbon 6, and essentially is a hexokinase. So it's a kinase enzyme. Now, just note that I've only drawn an arrow pointing one way, going in one direction, and this means that it's an irreversible reaction. But it's not always the case. For example, here, you will see that between going from your G6P, your glucose 6-phosphate, into fructose 6-phosphate, the difference is up here. So you got your carbonyls in here, and your hydroxyl groups from carbon 2 and carbon 1. Essentially, they are basically be swapped around. So you can see the functional groups in here are actually just swapped around from each other. That's basically all it does. Phosphoglucoisomerase will essentially just swap them around. Now, you will see that the arrow points in kind of both directions, which means that it's a reversible reaction. We can go from F6P to go into G6P and the other way around. So basically, they can be interchangeably made. Now, moving on to step 3. So essentially, we got your irreversible reaction again. So going arrow going one way, and you will see that it's conversion from F6P to F16P. Note that we have included the prefix bis. And what this means is that it's 2, basically 2 of something. And because it's next to phosphate, it's 2-phosphate. We have included another phosphate into the actual molecule. Now, so the phosphofructokinase essentially brings in another ATP, and this would then be made to ATP. Therefore, we have taking another phosphate from the ATP, dephosphorylated this ATP, and phosphorylated this molecule of F6P in carbon 1. And essentially, this makes the phosphofructokinase a kinase enzyme. And also, it's hinted here as well. So now we'll move on to step 4. So here So this is actually my favorite step. So what actually happens is that you will get your fructose 1,6 bisphosphate. Use the aldolase enzyme in this reversible reaction to essentially split this molecule down the middle in here between carbon 3 and carbon 4 and produce two molecules, your dihydroxyacetone phosphate, DHAP shorthand, and glyceraldehyde triphosphate, which is your GAP. Now, essentially, in order to progress from later stages, from going from these steps of energy investment, because so far we have invested 2 ATPs, but we haven't produced any ATPs, but we want that, we want to produce energy, but so far we have only added ATPs into the reaction. So how do we change that? Before going into the energy payback kind of stage, we need to convert your DHAPs into your GAPs. Okay, so this has to be converted to this. Now this is covered in step 5. So what I've basically done is kind of like taken the DHAP. I've basically put it in 180 degrees structure and made it this structure over here, and basically now it's a bit easier to see the differences between the GAP and the DHAP. Now, using the enzyme triose phosphate isomerase and using this reversible reaction, I've actually noted the differences and highlighted the differences between both GAP, DHAP, and GAP, and it's actually just between carbons 1 and 2 on both sides. Now it's just like a matter of like swapping them around, swapping the hydroxyl and carbonyl groups around. So you can see here, and then we will get to step 6. So the enzyme GAP dehydrogenase enzyme essentially is used to we use a phosphate molecule from the solution. So you can see here there's a phosphate molecule. This comes from the solution and not from ATP being dephosphorylated. So this comes in from the solution, comes into the reaction. This reversible reaction comes in, and also we will get a NAD plus. NAD plus would then get reduced into NADH plus H plus, and also this will also help us to oxidize the GAP molecule, this molecule. Now what happens is that this will note the differences. There's a phosphate and a oxygen, and here it's hydrogen. So in here, what happens is that the GAP is just converted into 1,3-bisphosphoglycerate. It's through phosphorylizing the GAP's carbon 1. So just this one. Now step 7. So just taking the same molecule as here, where it's 1,3-bisphosphoglycerate, and we'll then convert it to 3-phosphoglycerate. Not the difference is we have removed the phosphate. So this is leaving a negatively charged oxygen from here. So this uses the enzyme phosphoglycerate kinase. Now also we have, just note that we have used an ADP into an ATP. You can now tell that this is the energy payoff stage. We will now produce your ATPs because we have converted from ADP, which lacks a phosphate, remove this phosphate, taken this, put it in here, and then made an ATP. But note that this happens twice because we have, before we split the 6-carbon molecule into 2 3-carbon molecules, and now we have essentially produced, just one of the ATPs, which should essentially happen twice. So we have produced 2 ATPs so far. Now we have just we have paid off already the initial investment, but we want a benefit. So let's move on. So this is step 8. So it's a bit hard to read, but basically in step 8, not the difference, it's just like the middle carbons 2 and 3. So now we will swap around these molecules in here. So converting the 3-phosphoglycerate into 2-phosphoglycerate in step 8 with the help of a phosphoglycerate mutase enzyme, we will swap around the phosphate from the phosphoester linkage in here in carbon 3 with the carbonyl group from carbon 2. So we just swap this around, you will get this molecule, which is your 2-phosphoglycerate. Okay, now let's move on to step 9. So in this in step 9, basically we have here 2-phosphoglycerate being converted to phosphoenol pyruvate. Now this molecule, so this process step essentially uses the enzyme enolase, and as you can see here, the diagram includes removal of water. So what happens is that looking at the difference again between both molecules, you can see that the only difference is just the missing H2O, you know. Now, and also there's a double bond, you know, in an alkene between carbons 2 and 3. So double bond between the both carbons, and this forms a structure known as enol, which is an organic compound containing a hydroxide group directly bonded to an alkene, which is the carbon-carbon double bond. So this makes a phosphoenol pyruvate, which you can see here. But why would you want this? Well, actually, the phosphoenol pyruvate structure is more unstable and hence wants to break apart, which is perfect for the final stage. So we've actually prepped this to for the final stage, which will make your pyruvate. So finally, in step 10, we get this step, which is converting your phosphoenol pyruvate into your pyruvate. Now, this is the final molecule we want to aim to produce, and this is an irreversible reaction as well. Now, you will notice that we actually have produced another set of ATP. So this is, the second set of ATP that we'll produce. And essentially, this makes up, our energy payoff stage where we actually produce our ATP rather than being, in a balance or in a depth. So this results in production of pyruvate, which will be used in the process known as Krebs cycle. But I'm planning to cover this on another video. Anyway, thank you so much for watching. Please do some further reading on this topic because there's a lot of information that I haven't included, but it's really useful. There are some great websites out there that will provide you with a more in-depth knowledge about glycolysis. But anyway, thanks for watching this video and taking your time to understand some biochemistry.